Fine, and these are the quite some images of the few scanning electron microscope images of the pollen grains. So you can see there is a lot of differences in the structure of the pollen grains. And sometimes, as we are talking about the taxonomical studies, the systematic botany, we are going to consider the morphological characteristics for the classification. And later on, we have started with the numerical taxonomy, cytotaxonomy, palenotaxonomy. In that palenotaxonomy, we'll stick ourselves for the study of the morphology. Morphology, side, shape of the pollen grains can also be considered for, it also can be considered as a tool for the classification of a plant. Sometimes what happens, the plants may be having almost all the similar characteristics. At that condition, we can adopt the palenotaxonomy. So we can make the bifurcation, we can make them into separate two different species or a genera or anything according to the taxon. So in view of that, let me talk about the some information about or some introduction about the this one, that is the pollen structure. So as we already know that the pollen grain itself is not a male gamete because it is the only the precursor. We can broadly we can say it as it's a precursor because this particular pollen is undergoing the meiotic division and pardon the mitotic division and leads to the formation of the germ pore. I mean the germ generative cell. Another one is the vegetative cell. In the generative cell, we are going to get the male gametes, right? So the pollen itself is not a male gamete or it's not a sperm, but which is acting as a precursor for the production or a formation of a male gamete. Where we already know that each and every pollen will be having the two different cells. One is the vegetative cell, which will be the bigger one, and the generative cell, which will be the smaller one, and that is happening just because of why? Unequal mitotic division that leads to the formation of the two cells, where this particular vegetative cell will be considered as a non-reproductive cell. Why? Because it is not at all involved in the production of any of the gametes. The main role of the vegetative cell is to provide support as well as provide all the nutrition required for the formation of a pollen tube, because this particular pollen tube which is going to become coming out through the germ pore, and through the germ pore only we are going to see the movement or a transportation of the male gametes. It may be 2, 3, 10 or 100, anything it may be. But this particular vegetative cell which is acting as a guide or a navigator for the movement of these male gametes to reach the ovule. And this particular vegetative cell, that's what I already told you, that acts as a producer of a tube. That is what we call it as broadly a pollen tube. And through the pollen tube only we are going to see the movement of the components which are going to be synthesized in the sperm, I mean the generative cell. That is what the male gametes. Especially that particular male gamete is nowhere having any of the cells. It's exclusively the nucleus. Okay, now when study ಮಾಡ್ತೀವಲ್ಲ that it doesn't contains any components like ergastic substances, ribosomes, ಆ ಥರ ಏನಿರಲ್ಲ. That particular gamete contains only the nucleus, that is only the DNA. Apart from that, only the nuclear components will be there. All the cytoplasmic contents will not be seen in the male gamete. It may be anything, it may be sperm or it may be ovule. After the fusion of these male and a female gamete, what we are going to get the zygote, there we are going to see the further developmental stages. There you can see the formation of all the different components, a typical cell which will be having. Next, this particular pollen is produced in the microsporangia of the male cone of both any of the organism which are having said that, which can produce the male and a female gamete through the specialized reproductive structure. When it comes to the angiosperms, we are going to get the pollen grain from the androecium or a stamen. Where in case of in the gymnosperms, where we are going to get a male cone will be different and a female cone will be different. So nothing but any of the organisms which are capable of undergoing the reproduction through the sexual mode will have to produce these male gamete, and vice versa, that is a female gamete also. So some of the conifers we have, some of the gymnosperms where we are going to get the pollen grain as well as even in the angiosperm, because angiosperms are exclusively meant for the sexual reproduction. They are well adapted for the sexual reproduction, where in case of even the gymnosperms also, we can see some of the conifers, some of the pinus plants are capable of producing the pollen grains. And that's the last one is the important one, as we have seen in the previous slide, that is the shape, size, and the composition as well as the surface making. The structure, sculpture as well as articulation and the shape or the surface of the pollen grain is quite distinguishable. We are not going to see the similar shape, similar size, similar structure as well as similar morphology in generally among the different species, different genera, different family, where each and every species will be having its own unique pattern of the pollen grain. And that's what the more important or significant marking characteristic of a pollen. And now coming to the pollen wall. So we already know that broadly the pollen wall broadly contains the two, outer one is the inner one layer. Outer one is the exine layer and inner one is the intine layer. So till now what we know about the pollen wall is exine as well as the intine. Further, this particular pollen wall is the external wall, the outer wall which is stratified. This particular stratified nothing but we are going to see the ridges and furrows which is not uniformly distributed. The composition what we are going to get secreted from the tapetum which is not going to be deposited uniformly on the surface of a pollen. Where exine which we, because of the uninterrupted depositions of the secretion received sent by the tapetum, where we are going to see the irregular depositions on the exine. Because of that we call it as a stratified wall, where this particular stratified wall we are going to see only in the outermost wall. That's what only the exine. That is the anther wall, broad, that make it as a pollen wall, not as the anther wall. The online students, ಅಲ್ಲಿ second point AW ಅಂತಿದೆ. Okay, it's not
Yes, sir.
anther wall, it's a pollen wall. Okay na, changes ಮಾಡ್ಕೊಳ್ತಾರೆ. So the particular pollen wall consists of, we know that the basical components, one is the exine as well, another one is the intine, where the exine which will be having sculptured characteristics because of the irregular distribution, irregular deposition of the substances released from the tapetum, where intine is quite smooth enough, where we are not going to see the variation in the ridges, where we are not going to see the variation in the thickness of the entire intine region. The uniform distribution, that's again the cellulosic matter will be there, and that particular cellulosic matter will be uniformly distributed on the intine region. And this particular exine further structurally, which is divided into two types, one is ectexine as well as the end exine. There you can see in the right side of the slide ectexine as well as the end exine. The structural classification of exine into two types, so two subtypes and karibu ectexine and end exine. Again ectexine ಅಲ್ಲಿ ಮೇಲ್ಗಡೆ ಎಕೆಟೆಕ್ಸೈನ್ ಆಗಿದೆ, ಓಕೆನಾ, ಒಂದ್ಸಲ ಕನ್ಫರ್ಮ್ ಮಾಡ್ಕೊಳ್ಳಿ ವೆದರ್ ಇಟ್ ಇಸ್ ectexine or ecetexine. Fine. The online students also make it clear that whether the outermost layer in the exine, whether it is ecetexine or the ectexine. Fine.
Yes, sir.
Okay. So outermost layer of with this particular exine which will be having, let us take which is around 2 micrometer or 0.2 micrometer, whatever it may be, that particular thickness, in that particular thickness we are going to see the structural variation in the layer. That is what, where we are going to see the first layer, the outermost one is ectexine, the innermost one is a endexine. All these two layer together will be considered as the exine. This is what, based on the structural differences, or structurally we can classify the exine into two types, where this particular ectexine, next based on again further deeper classification based on the composition as well as the structure, the whole pollen wall is classified into three different layers. One is a tectum, columella and the nexine. Fine. Only the exine structurally classified into two types, one is ectexine, another one is the endexine, where further based on the composition as well as the structure, the whole pollen wall is classified into three layers. One is tectum, columella and the nexine. These two, tectum and columella, are the parts of sexine. ನೀವು ಮತ್ತೆ ಹೇಳ್ತೇನೆ ಏನಂದ್ರೆ sexine and nexine ಅಂತ ಬರ್ತದೆ. Sexine which is a outermost layer of exine. Next, nexine is the internal wall of the exine. This nexine always uniform distribution ಆಗಿರ್ತದೆ. ಸೊ ನಮಗೆ ಕನ್ಫ್ಯೂಷನ್ ಆಗಬಾರ್ದು ಅಂದ್ರೆ ಇನ್ನ ಈಸಿಯಾಗಿ ಹೇಳ್ಬೇಕಂದ್ರೆ pollen exine, don't make it as a pollen wall. Exine is divided into two types, one is a sexine as well as the nexine. This sexine which is the sculptured one, because of which is outermost layer, because of a deposition what we are going to see it from the tapetum and the outermost layer, where we are going to see the sculptured or articulation as well as the stratified wall on the sexine, and that region will be considered of that particular exine region will be considered as a sexine. And next to the sexine what we are going to get, which is quite uniformly distributed layer, there we are not going to see any articulation, any stratification and any of the sculpturing characteristics. That is what the nexine. ಅದಕ್ಕೆ ಇಲ್ಲಿಂದ 1 2 3 4 and 5th statement that will depicts about the exine, which is divided into two sublayers. One is sexine as well as a nexine. Non-sculptured one is the nexine, which is quite distinguishable, which is just found between the intine as well as exine. Exine is entilkore. This particular exine, outermost one is exine, next nexine, next intine. Fine. Now, this particular exine, when we talk about the complete pollen wall, tectum and columella are the two important components which will be considered as the parts of a sexine. Why? Because you can see in the image that in the tectum as well as in the columella, you can see there is an distinguishable boxes are there, just like a vacuole-like structures are there, and you can even see some of the dots, right? The dots are like, so those dots which will depict about the articulation or stratification. And these two, tectum as well as the columella, belongs to the wall, that is a sexine, nothing but which is again a pollen exine outermost wall. And next, in the nexine, what we are going to get? Again, here we are not going to see any such stratification, all those things, which is the innermost layer. And again, this usually in the E image, you can see the nexine one articulation is stratification. But when it comes to the theoretical aspect or practical aspect, we are going to see the both nexine 1 and the nexine 2. These two are completely the uniformly articulated layer, where we are not going to see any perforations or where we are not going to see any stratification. And next to the nexine, what we are going to get, the innermost wall is the intine. So the pollen wall is having this particular pollen wall is broadly divided into two walls. One is the exine as well as the intine. This particular exine, which is again based on the structural characteristics, we have classified them into ectexine as well as the end exine. This particular ectexine, which is again further classified into tectum, baculum, and a fruit layer. ಇಲ್ಲಿ columella and baculum both are same. Both in the fourth point where I have given it as ectexine, tectum ide, baculum ide, and fruit layer ide. This particular fruit layer is nexine 1 antidella. Nexine 1 is the fruit layer. The columella is the baculum. Sometimes you may ask, these two, nexine 1 and nexine 2 are the parts of the nexine. Just see on the right side in the ectexine, tectum, columella, fruit layer. These three are together will be considered as the ectexine. Where is nexine 3, tectum, columella, nexine 1, 3 will be considered as ectexine. You just compare that left side with the right side image. Part of the nexine wall will be the part of the ectexine. The online shows exine, which is a peripheral wall or outermost wall of a exine, and in next we have the nexine, which is the layer which is found between the sexine as well as the intine. Together, sexine as well as the nexine will be considered as the exine. Is that clear? Should I repeat anything here? Is it confusing? No? Where the main chemical composition of a sexine is a sporopollenin as well as leaf bodies. Fine. Where intine and nexine, where we are not going to see such a deposition because which are inner to the exine, nothing but inner to the sexine only. We are going to see the pectin components as well as very limited amount of pectin components as well as a chief amount of the cellulosic components will be there. It may be a cellulose or it may be hemicellulose. Pollen wall, and in this particular pollen wall, we'll talk about some of the intra aspects about the chemicals, pattern, structure. Where we, as I already told you, that the intine, which contains the pectic as well as cellulosic components, that particular pectic as well as cellulosic component may be individually distributed or they may become complex structure and they can be available in the intine. So the pectin may be available individually, cellulosic component may be available individually. Sometimes the complex structure of these two, the pectocellulosic components may be there, and all these three, either individual or in composition, will be served as the primary wall for the most of the intine. It is here, it was given as somatic cell. Still the pollen is somatic cell because it has not yet undergone the production of a gamete. Fine. Now the cellulosic component, microfibrillar structure, I think we have already discussed in the theory class or online class anatomy cell wall from cellulose, from microfibrils, measles, cellulosicum. That's what the microfibril structure, microfibrillar pattern of the cellulosic components will be there in the pollen wall, and the structure of the particular this intine maybe, sorry, not the intine, this particular structure of the pollen wall may be bead-like structure, or a ribbon-like structure, or a plate-like structure. These three structure are going to be released by, are going to release a specialized enzyme for breaking off the wall. Why? Because once the pollen is coming on the surface of a stigma, it has, the germ pore has to come outside. Germ pore come ಹೊರಗಡೆ ಹಾಕಬೇಕಾದ್ರೆ it has to search for a, this, I mean, the vegetative tube has to come and it has to search the germ pore. And near the germ pore, we are going to see a different structure on the pollen wall. It may be a bead-like structure, ribbon-like structure, or a plate-like structure. These three generally ಹೇಳ್ತಿರೋದು. Most of the structures ಏನಿರ್ತದಲ್ಲ, majority of the pollen wall will be having bead, ribbon, or a plate-like structure. And these three, if they, whatever it may be the structure of that, the respective enzyme is going to be synthesized according to the composition. If enzyme ಜಾಸ್ತಿ the component ಕಡಿಮೆ ಇತ್ತು, enzyme ಜಾಸ್ತಿ release ಮಾಡಿ is of no use. The plants are very economic. They are not going to release such any adverse, any extra molecules for the breaking down. Sometimes ಏನಾಗ್ತದೆ, because of some adverse condition, the cellulosic component or a pollen wall or a germ pore which maybe looks quite tough enough. At that time, enzyme release kadime aitare, it's quite difficult. And all the enzymes, we already know that the substrate as well as enzyme has to get attached each other. And only when there is agglomeration, a standard optical agglomeration, then only we are going to see the breakdown of the substrate. Similarly, the bead-like, ribbon-like, and plate-like structures are going to release a different types of the proteins or enzymes, and they are available at the near to the germ pore. Why? Because these three different structures will release a specialized chemical or enzyme which will allow the components which are present on the germ pore to break down, and only the germ pore, the vegetative tube has to come outside through the germ pore. Next, exine is chiefly composed of the, that's what the class material sporopollenin, and this particular sporopollenin is formed by the fusion or a complex structure because of the substance released from the tapetum as well as pro-ubist bodies. And it also in most of the articles it has been mentioned as a class material, because we are not yet clearly understood what is sporopollenin because of that. And this particular sporopollenin, which is derived from the carotenoids by oxidative polymerization. And why this particular point is nowhere related to this particular slide, but when I'm searching for this one pollen wall, I thought I got this particular statement. This sporopollenin ಏನು statement ಇದೆಲ್ಲ ನಿಮ್ಮ sporopollenin ಎಲ್ಲ ಹಾಕೊಂಡ್ಬಿಡ್ರಿ. Fine. It has been said that the sporopollenin is derived in most of the plants. This from the oxidative polymerization of the carotenoids. Fine. And this particular sporopollenin is quite resistant to the heat, temperature, physical as well as biological substances, and which is not going to be broken down at all. This particular pollen wall, this the protective function during the hazardous journey during pollen movement. That's again the function or the scope of a pollen wall is that it has to be transferred through the wind, I mean the abiotic factor or the biotic factor. At that time, it has to get exposed to different hazardous conditions. So to overcome that, to protect the internal structure, the pollen wall is going to be developed. And the different pattern of exine will be having its own taxonomic value. That's what I have stated in the beginning of the today's class, that each and every pattern, each and every morphological changes, each and every size of a pollen grain can also be adopted as a tool for classification of a plant. This particular pollen kit, usually the composition, again a specialized chemical composition, what we are going to see it on the pollen grain is generally will be considered as a pollen kit. This particular pollen kit is a viscous substances. Usually we are going to see it on the pollen grains which are going to be pollinated by the insects. The insect pollination again, Anemophilica, entomophilic, right? Right. The pollen, the oily layer which is forming a thick viscous component on the pollen grain, where the insect pollinating agents or insect pollinating pollen grains will be contain the pollen kit. Nothing but the accumulation of pollen kit is more on the pollen grains which are usually pollinated by the insects. And you may know, you may ask, so why that particular pollen, such composition can be seen on the pollen grain? Because the insect has to get attracted, and they have to get attached to the surface of a body of a pollen, I mean the insect. So because of that, this particular composition, again, adaptations, morphological variations, we can see to again to have a growth and development and to increase their population. And this particular kit, pollen kit, the oily layer may be sticky or may be having some odor, it may be a colored also. And these three characteristics also more important in the insect pollination. Stickiness, it may get attached to the body of a pollen, the insect, and odor as well as the color is to attract the insects for the pollination. Because of the presence of a pollen kit only, these three, what we call, these three important characteristics we can see it in the pollen kit. And these again, these three different different composition of the pollen kit is also served by the tapetum itself. Let me make you clear that all the components of on the surface of a pollen and a pollen wall is completely generated by the tapetum itself. That's what will be considered as a nutritive as well as a nourishing layer or nourishing wall. Fine. Next, it is composed chiefly of carotenoids or flavonoid pigments. We already know that the pigments which are having their own color, usually the chlorophyll green, carotenoids and the flavonoids. Okay, the carotenoids and the flavonoids which will falls between the yellow, orange, and red color. Okay, na? And because of this particular, the odor as well as the color is due to the presence of the flavonoid as well as the carotenoids. This particular carotenoid will give you the color around between yellow, orange, or red, or even the pale red also. And the flavonoids, which are right again, these are the secondary metabolites which will emit some odor. Fine. ನಿಮಗೆ ಟೀ ಮಾಡೋ flavonoids ಸ್ಮೆಲ್ ಬರುತ್ತಲ್ಲ, that's what the flavonoid, because of a flavonoid rich antioxidant rich. The carotenoids as well as the flavonoids are the chief compounds. I mean, they are only two compounds antalla. The major components of the pollen kit are the carotenoids as well as the flavonoids, and that is what the characteristic yellow or orange color of the pollen grain. And the cementing mechanism, nothing but it has to be the jelly-like structure. It has to be, because to get attached to the body of an insect, it should be jelly-like. It should be sticky. So for the particular cementing action of a pollen kit, we are going to see the presence of a glycoproteins. You may know already that glycoproteins, nothing but glycoprotein. Usually glycoprotein. Have you studied fluid mosaic model? They have studied about the membrane. Hydrophilic as well as hydrophobic layers. These are again like glycoproteins we can see, and the lipids we can see, the glycolipids we are going to see, and the monosaccharides. And these monosaccharides may be the glucose or fructose, right? And these pollen kit, the main function of a pollen kit, apart from contributing themselves for the insect mediated pollination, and they are going to protect the DNA, they are going to protect the chromosome, they are going to protect the internal nuclear material from the UV damage, because these particular pollen grains are quite, which will be having, if they get this particular sporopollen has lost its existence, then they become the naked pollen grain. These naked pollen grains they may get exposed to the UV light and they may get damaged. So to avoid that one, this pollen kit also renders themselves for the UV damage. I mean, to avoid the DNA break. And insect attractant. That's what, because of the main role in the formation of a pollen kit is to attract the insect to come up with the pollination, entomophilic pollination. And it is also act as adherent to this insect body. Again, that's also completed. And this is quite hydrophobic in nature. Because of hydrophobic in nature, then they can go attached to that. If there is any possibility of hydrophilic property, then they may get solubilized. Once if they get solubilized, then that particular pollen will not be having any role to get attached to the, they won't produce the color and they won't produce the odor. So because of hydrophobic characteristic, they are quite long lasting and they can bring about the insect pollination. And this is quite associated with the dispersal of a pollen grain. And this image which is depicts about the a mature pollen grain containing a thick outer wall sculptured again. You can see the sculpture is either the ridges as well as the furrows, as you can see here. This one, this is the pits. This is what the outermost one. Then next we are going to see the inner one, which is quite uniformly distributed, right? Okay, and this is what pollen tube nucleus. Again, nothing but a vegetative cell nucleus, and another one is the generative nucleus. Later, this genital nucleus will undergo the mitotic division and will form the two male gametes. And this is what about the today's class. Is that clear?